So my research group is involved in the study of physiological and pathological angiogenesis, in particular the study of vascular endothelial growth factor family. And the data that I'll show you today may be defined as a gain and loss of function of a vascular endothelial growth factor receptor 1 by the overexpression of its specific ligand, placental growth factor, in tumor context, by showing data what we obtained using a specific anti-FLT1 peptide that we identified. So as Raf showed this morning, there is a long list of diseases in which there is increased or decreased vascularization. And so the therapy until now, anti-angiogenic therapies for cancer disease and age-related macular degeneration. But it's important to underline that recent data from CPD of Atlanta indicate that about 1/3 of the world population may impact with inadequate or exuberant angiogenesis. And this is a list of anti-angiogenic drugs approved in clinical trials. All are directed against members of vascular endothelial growth factor family or related receptors. And this because this family play a crucial role, as you well know, in vessel formation. It is well known that the block of VEGF-A and KDR is crucial for, in particular, for the activity of this axis on endothelial cells. But it is also well known that the block of placental growth factor or vascular endothelial growth factor receptor 1 give an important inhibition of angiogenesis. In many preclinical models, it has been demonstrated. And this because the activity, the block of this axis is important not only for the activity on the endothelial cells, but also because it's crucial for the recruitment of many different kinds of cells that has a role in angiogenesis, such as BM progenitors, in particular inflammatory cells, monocyte macrophage cells, smooth muscle cells. In addition, recently it has been reported that an upregulation of VEGFR1 specific ligands after block of VEGF-A by pharmacological therapy, so indicating that this may be an alternative way for tumor to grow. So concerning the VEGF family, and this seems it's important for me also to introduce the first data that I will show you. So normally you can see on each review that you read on vascular endothelial growth factor family that VEGF-A may bind the two receptors, R1 and R2, PIGF and VEGF are specific, but in addition VEGF-A may also induce receptor heterodimerization. But very often it's forgotten that if cells co-express VEGF and PLGF, heterodimer may be generated. And these heterodimer are able to interact only with the receptor 1 or induce receptor heterodimerization. Thus, in this condition, we practically move the activity on receptor from KDR to receptor 1. And in addition, recently it has been also reported gremlin, another protein that is able to interact specifically with R2. And it is well known that exists also soluble receptor 1, and this is one of the most potent anti-angiogenic molecules because it is able to prevent the activity of all the possible dimer. And also soluble KDR that in soluble form is, seems to be more specific for VEGF-C rather than VEGF-A. So the mechanism of heterodimerization have to be considered in the next experiments that I'll show you because we overexpress PLGF in tumor cells. We did this by stable clones, tumor cells, stable clones, or by gene therapy. I'll show you the data also of delivery of PLGF by gene therapy. And we have also generated the variant of PLGF named the PLGF-DE. This variant loses the ability to bind and activate receptor 1, but is still able to generate heterodimer. So we have two different conditions, inhibition and wild-type PLGF. These proteins are enabled to bind to any receptor, so in this condition we lower the concentration of VEGF-A, forcing the formation of the heterodimer. In that condition we lower the concentration of VEGF-A, but we have active PLGF homodimer and the active PLGF-VEGF-A heterodimer. So when we quantify the dimers As you can see, we were able to lower the concentration of VEGF of about 50% in both cases, so using the PIGF wild type or our variant. We have the appearance of the heterodimer and, of course, the overexpression of PIGF. So we use it as a tumor model R2780 cells, is an human ovarian carcinoma cell lines, and we delivered PIGF wild type, PIGF-D, or as controlled GFP by gene therapy. This is just to show you that the cells are susceptible to the infection of the adenoviral particles. And we injected cells, and then when tumor reached about a volume in the average of 200 milligrams, we inject intratumorally the virus, and after 7 days we repeat the injection. So what we observed was a very strong inhibition of tumor growth using our PIGF-D variant. So when we have lowered the concentration of VEGF and generated inactive dimers, while the overexpression of PIGF wild type did not give any difference compared to the control. But what happens in terms of vessel density? So as you can see, we did not got any difference, statistical difference between PIGF wild type and control. Of course, in the case of inhibition, we got a strong inhibition. But when we analyzed the vessel based on their dimension, we observed that the overexpression of PIGF wild type determined a reduction of small vessels and an increase of larger vessels. Moreover, when we analyzed the inflammatory infiltrate, in particular the F480 positive cells, we observed that with overexpression of PIGF, we got a very strong increase of monocyte macrophage infiltration. And at the same time, also when we evaluated the vessel stabilization by covering of vessel by smooth muscle cells. So the overexpression of a specific ligand of FLT1 induces a very strong recruitment of inflammatory cells and an increase in vessel stabilization thanks to the activity of recruitment of PIGF-FLT1 axis of these cells in the site of angiogenesis. So now let's move to the story of peptide. I want to show you that we have identified, we have screened different combinatorial peptide library. We have a longer experience in this by a simple ELISA by the same which the inhibitors as used as competitors of the PIGF-FLT1 interaction. And from the library screened, the tripeptide tetrameric library was the best in terms of results. This particular structure, that means that we have 4 times the same sequence assembled on a core of lysine, because these molecules are extremely flexible, no conformational preferences, may adapt on a high recognition surface. In addition, when we synthesized the library, we used a natural amino acid, 30 unnatural amino acid. So we have a library composed of 27 times peptide. Here you can see the list of amino acids are all the amino acid and some chemically modified amino acid. And the 3 rounds of screening allowed us to identify diglutamic acid, L-cysteine S-benzyl amino acid, and L-cyclohexylalanine amino acid. So the schematic structure of the molecule we identified is this one, and this is the chemical one. So the screening, we don't know if the peptide selected bind PLGF or bind to the receptor, but we immediately do dose-dependent experiment to verify if this molecule is able to inhibit also VEGF-A FLT1 and VEGF-B FLT1 interaction, meaning that probably peptide bind to the receptor. And this was the case. We have a very nice dose-dependent experiment for PLGF FLT1 and VEGF-A FLT1 interaction with an EC50 around 8-10 micromolar. And the molecule was also able to inhibit VEGF-B FLT1 interaction, also if this C50 was a little bit higher. So next, we confirm that molecule bind to the peptide. This is a dose-dependent binding. It is interesting to see that the affinity is higher for human receptor. It did not bind to mouse, to FLT1 or KDR, so to the receptor 2. This is also confirmed by phosphorylation experiments, the peptide is able to inhibit in a dose-dependent manner the activation of FLT1 stimulated by PLGF or VEGF, but did not affect the phosphorylation of KDR on UVEC cells stimulated by VEGF, and is not able to inhibit an interaction between PDGF-B and PDGF-B receptor beta. So we start to test the molecule in cell-based assay. This is a very important thing. We use the natural amino acid, and the molecule is really stable. After 7 days in serum, the molecule is practically untouched. So we do a classical capillary lactube formation. We have a full prevention of capillary stimulated by PLGF or VEGF using the molecule at 20 micromolar. And here you can see two different controls that are not able at higher concentration to inhibit the formation of capillary. And then the first assay that we did in vivo, it may appear as a paradox, is an assay in which we stimulate vascularization, but this may happen only in the cornea, because in the cornea it has been described a very molecular peculiar condition. So the cornea expresses VEGF-A, also if it is avascular, and the avascularity is maintained for high expression of soluble receptor 1. It is not expressed full-length receptor 1. So if we inject in cornea anti-FLT1 molecule, for example, an antibody anti-FLT1, we were able to displace VEGF that become free to act on the vessels of the conjunctiva, and the cornea become vascularized. So in physiological condition, this was a fantastic platform to test our peptide. And indeed, those increasing concentration of peptide injector corresponded an increased vascularization of the cornea. And this means also that peptide also in vivo do not interact with the receptor 2, otherwise these vessels are not formed. So we move on a tumor model, but we have to solve a problem. Our peptide is not soluble, the solubility is lower in saline condition, so we tried different conditions, different adjuvants, and at the end we found that a solution of PEG 400 water 1:1 gave us a very nice suspension, very stable suspension, and injectable suspension. Then we start the assay on tumor, first on syngenic model of colon cancer using CT26 cells, and the condition of delivery of the drugs was here indicated. We used the peptide at 50 milligram kilo, considering the EC50 in vitro, and each today considering the strong stability the peptide show in serum. We compared the assay with antibody anti-mouse PLGF kindly provided by thrombogenics. And as you can see, our peptide give a very strong inhibition of tumor growth, significantly lower compared to anti-PLGF peptide. So we start the analysis of the tumor. The weight of the mice did not change during the treatment with our peptide. We analyzed also the peritoneum after the sacrifice of the animals, and any gross change of toxicity were observable. And when we analyzed the vessel density, we obtained an inhibition comparable between the anti-PLGF and our peptide. But when we analyzed the F480 monocyte macrophage infiltration, we got a very strong inhibition with our peptide. So we moved to a human model, and in this case we compared the effect of peptide to that of Avastin delivered at 5 milligram kilo, and anti-human PLGF kindly provided by thrombogenics and delivered 2 times a week at 25 milligram kilo. And it was really amazing to see that our peptide practically has an activity fully comparable to that observed with Avastin, and it worked better than anti-PLGF antibody. And of course, the reduction was significant compared to our control. So we determine vessel density, and as you can see, we got about similar results with the 3 drugs analyzed. And of course, you can see representative pictures. And when we analyze monocyte macrophage infiltration, we got a very strong inhibition with our peptide, significantly lower compared to Avastin and compared to anti-PIGF antibody. And in that case, we analyzed also the vessel stabilization and the peptide give inhibition similar to that observed with the two antibody tested. And this is a very important data. So the peptide is effectively able to inhibit FLT1 phosphorylation in vivo. So we prepare tumor extracts and we mixed the equivalent amount of extract from mice of the same group, and then we did Western blot analysis. So we got a partial inhibition using anti-PIGF or Avastin antibody, and we got very, very strong inhibition when we delivered our peptide. So next step was to verify if we can lower the dosage of the peptide. So we did a dose-dependent experiment again on human colon cancer xenograft model made using HGT 160 cells. And as you can see, we give two different dosage, 25 milligram kilo and 10 milligram kilo, and at 25 milligram kilo we got the same results of the previous one, while at 10 milligram kilo practically there was a little effect that was not significant compared to control. So we were able to lower the dosage at 25 milligram kilo. And next one, we verify if it's possible to obtain synergic activity with chemotherapeutic agents. The current therapy for human colorectal cancer are the 4-fill combination that is composed by 5-fluorouracil, irinotecan, and oxalylplatin. So we decided to compare the activity of our peptide and to try a combination with irinotecan that was delivered at 50 milligram kilo 1 time a week. So what we obtained was this. Again, our peptide give an inhibition similar to that of the Avastin, and the irinotecan alone give similar inhibition, but the combination was striking. We observe a very strong inhibition. And at 28 days, in which only 2 of out of 8 animals of the group, the tumor was still detectable. Then at that point, we stopped the experiment for all the groups. And what happens for the other animals with the double treatment, we here stop the treatment. That tumor was not detectable for about 3 weeks, and then after it starts to regrow. So this data indicated, of course, that the combination, the synergic activity is possible. This is this is just the first experiment, so we have to confirm the data and to repeat and to verify other possible combination. So also in this case, we did not observe significant change of the mice weight except for the 2 groups treated with irinotecan, but this is expected. The loss of the weight was about between 10 and 50% of the original mice weight. And once again, we analyzed the phosphorylation, and we still obtain with Avastin a partial inhibition and a very, very strong inhibition in the mice treated with our peptide or with the combination. And finally, you well know that Avastin is also delivered intravitreally for age-related macular degeneration therapy, and so we decide to verify if this peptide may be active also in this disease model that is generated by generating damage on the branch membrane by laser. And what happens is an overgrowth vessel from choroid versus retina. And we performed the experiment in this condition, 6 animals per group, 20 laser spots per group. At time 0, we did the injury, and we injected intravitreally, so as normally happens in therapy for human, 50 micrograms of peptide in 1 microliter of total volume, and the anti-mouse VEGF-A at a standard condition. And the analysis was performed after 7 days. And as you can see, we got a very strong inhibition also of choroid neovascularization in a dose-dependent manner. At 50 microgram, the decrease was very impressive, minus 72.5%. This is practically the limit of the methodology. And then this was very nice results because if we expect something of important in tumor model, it is not what so expected also in choroid neovascularization model. So in summary, we have identified this peptide that practically confirm that the block of FLT1 receptor determine a strong inhibition of vessel formation, in particular of inflammatory infiltrates. That this peptide give a syngenic inhibition activity with irinotecan, one of the chemotherapeutic agents currently used for human therapy of colorectal cancer, and that peptide inhibit also choroid neovascularization. So let me thank two crucial collaboration for the project, Dr. Ruvo, that is the chemist that think and design and synthesize the peptide library, and Professor Ambati for the collaboration on the experiments on cornea and choroid neovascularization. The two agencies that grant the project and all the people of my group, in particular Laura Turisco and Valeria Tarallo. And important, all the ICGEB animal house staff that give us an important support for the in vivo experiment. And before to conclude, let me invite all you to the next meeting that will be held in Italy on angiogenesis in the fantastic Capri Islands, in the meeting in which we have which there is talk on cell signaling, on the role of inflammatory cells, and also on the two disease model will be cancer and ocular neovascularization. For people outside from Italy, we have 15 young travel awards granted by FEBS, 10 for European, and 5 are transcontinental. So ask to your young PhD student and postdoc to apply. Thank you.
Thanks a lot.